Okay, so in this video, we're going to talk more about the control of the cell cycle. This is of interest to us because of its really strong relevance to cancer. Cancer biology is all about how cells are controlling their normal behavior and how that normal behavior is sometimes lost. All right, and so I want to go directly over to the share with the textbook, but it's going to be the picture is going to be the cell cycle, and I guess before we get there, let me just identify a few things. We're going to be seeing three distinct checkpoints in the cell cycle, 3 points at which the cell cycle can be stopped. You're thinking, what this means is that there's an inherent tendency for the cell cycle to move forward. The only time that it doesn't cycle forward is when it's stopping at one of these checkpoints, right? And so there are 3 of these checkpoints. Remember that interphase has a G1, a gap 1, an S synthesis, and a G2, a gap 2 phase, right? And there's also the different stages of mitosis, in which you've got prophase, prometaphase, metaphase, anaphase, and telophase. And the metaphase is going to be the place where we have the M checkpoint. M stands for metaphase. And so what's going on are really kind of understandable things, right? And so it's more than just memorizing G1 checkpoint, G2 checkpoint, M checkpoint. What you really want to do is to say, well, why do these things make sense? Why is it a good idea for us to stop, for example, at the end of G2? Why is it a good idea for us to stop at the metaphase point? Okay. How does the G1 checkpoint affect whether the cells are going to be continuing the cell cycle or not? Okay. Keeping in mind that sometimes we want more cells and sometimes we really want cell divisions to stop. Okay, so on to the textbook. And there you have it. This is the cell cycle. Recognize it because we've talked about that in the previous one. We've got interphase, that's the tan regions. The mitosis, or the mitotic phase, is the part that's in blue. It's labeled M. And what we're saying here is that the cell cycle, the cells are basically continuing forward. They basically move forward. If they keep on going forward, right up until they reach the point where they're hitting one of these checkpoints. Okay? It really is kind of like one of those bars that come down across a railway crossing. Are we allowed to go forward? No, we're not allowed to go forward until we provide our credentials, until it's time for us to go forward, right? And so the G1 checkpoint is here, obviously at G1. G2 checkpoint is right there at the very end of G2. And the M checkpoint is at the metaphase point in mitosis, right? And so really quickly, it kind of makes sense. Remember that the big question that we've been talking about before is, do we need to continue the cell cycle or do we need to stop? Do we need more cells? If so, we let the cell cycle keep on going, right? We would keep on going into the S phase, then on the G2 phase, and then it's going to stop at the G2 checkpoint. But once we make it past the G1 checkpoint, there's nothing that's going to stop the cell from completing the rest of the cell cycle up until the time it reaches G2, the G2 checkpoint, right? And so right here, this checkpoint here is the point which somebody is going to be coming out and saying, well, do you have a license to move forward? Do we have the appropriate signals in place that validate the need for more cells? For example, in the case of the anchorage-dependent inhibition that we talked about, anchorage-dependent growth that we talked about in the previous video, do we have information that there is additional real estate, additional space that needs to be filled If that's the case, then sure, keep on going. We need more cells, keep on going, right? If not, okay, okay, then we stop the cell cycle. Stop the cell cycle, and we go into this thing called the G, I think it's the G0. But this is, we actually use the British term for zero, which is not, right? So if I refer to the G naught phase, that means this state in which the cell is basically stuck right here in the G1 stage, right? And so your body is composed of bazillions of cells. Most of those cells are in the G naught phase. They're basically told not to divide any further. Right? Sometimes we can take the cell out of the G naught phase and put them back into recirculation, in circulation, if we need to have more cells because maybe there was some type of lesion, right? Your skin normally doesn't need to continue to grow, at least parts not on the outside. If you were to cut yourself, you would have to fill in the gap with new tissue, and that's going to require more cells. And so that would be an ideal situation, an easily conceived situation in which some of those cells that were in G naught would be allowed past the G1 checkpoint, and then they go into the S phase, and then they continue on through the cell cycle, right? But yeah, this is a point in which we can either take a left turn and go into G naught, or we can continue forward and go into the rest of the cell cycle, right? So when it comes to the G1 checkpoint, we're really asking, do we need more cells, or do we want to stop the cell cycle? This is like our main point, in which we stop the cell cycle because we don't need any more cells. The M and the G2 checkpoints are going to stop the cell cycle, but not because we might need to arrest the cells from further growth, at least not usually. Right? The G2 and M1 and M checkpoints have other ways of rationalizing them. Right? For example, right? We know that mitosis, this whole song and dance of mitosis, requires a whole lot of things to line up exactly perfectly. We're going to make the chromosomes do this thickening. We're going to have all of the chromosomes line up on the metaphase plane. We're going to have them divide. And this is like a pretty serious endeavor. And if we're not ready to jump into mitosis, that could cause problems, right? And so it kind of makes sense. Mitosis is a big step. I mean, this is a big commitment to go past this point and into the mitotic cell division. We want to make sure that we're not jumping in mitosis before everything is ready to go. Okay. Yeah, I'm totally oversimplifying this, but that's the bottom line reason why we would have a G2 checkpoint right before mitosis begins. And only when we can assure ourselves that we have a state of readiness to launch into the mitotic division, that's when we're allowed past the G2 checkpoint and we go into mitosis, right? So yeah, a G2 checkpoint says, are you ready for mitosis? Yes. Well, then proceed on forward, right? We have no reason to stop you if you're ready to go in mitosis. But if you're not ready, hey, hang out here until all of your ducks are in a row, all of your growth factors are lined up exactly the way they need to be, everything is perfect, and then we can move into mitosis, okay? G2 checkpoint, ready for mitosis. Okay. The M checkpoint is even easier to figure out because if you looked at the pictures from that portion of the chapter that I didn't illustrate, we know that at some point we've got these sister chromatids that are lining up like this, right? These chromosomes with sister chromatids are lining up like this. And then we have the mitotic spindles that are attaching centromeres to one end. Here's the centriole on one side. We've got another mitotic spindle that's attaching the centrioles, that's attaching the chromosomes to the other side, right? And once we have anaphase initiating, right, these sister chromatids are then going to be, oops, let me do that in blue again. The sister chromatids are going to be pulled toward the centrioles in the opposite directions. In other words, this sister chromatid is going to go here, the sister chromatid is going to go there, the sister chromatid is going to go in that direction, the sister chromatid is going to go in that direction. This is what's required in order to get exact separation, perfect replication of the two cells with identical genetic information on both sides, right? Okay. Now, in the process of setting up this whole bit where you got mitotic spindle, you've got the attached spindle fibers, the non-attached spindle fibers, things have to be exactly right, right? And if things aren't exactly right, you don't want to proceed into anaphase, right? And then so, yeah, the M checkpoint is really easy to understand. Somebody is making sure all the chromatids are hooked up properly to their spindle fibers. All the spindle fibers are attaching to the correct positions. And then, and only then, are we allowed past the M checkpoint to go into anaphase, right? So think of the M checkpoint as saying, well, are you ready for anaphase? Right? If everything is perfect, move forward, please. We'll let you go right into anaphase, and then we go into telophase, cytokinesis, and then we keep on going. So the two cells will keep on going in the through G1 until they reach the G1 checkpoint. Then another century is going to stop them and say, hey, do we really need more cells? No, go into G0, yes, continue on past and go into the S phase, right? And so, what we're doing is I'm giving you what we call a model for this control, and there's a lot of empirical support to say that this is approximately how we have normal regulation of the cell cycle. This is what cells normally do. Okay, and sometimes cells don't behave entirely correctly, and that's the situation where we start seeing behaviors like the formation of a tumor, right? So if cells were supposed to go in this direction, but instead they were allowed into the S phase, then we would basically be continuing on with more and more cell cycles, and that's going to cause us to have a big lump of cells where you don't really need a lump of cells, right? That would be the formation of a tumor, right? And in truth, there are multiple things that have to happen in order for a tumor to develop into a full-blown cancer. But that's a discussion that we don't really need to have at the very moment. Okay. Actually, we'll get into some more of this later on in today's discussion. Okay. So we've done that. Checkpoints. We understand all of those. Okay. Now, the thing about these checkpoints is that if you think about, well, what could happen if we have problems? If we were to allow the cells to move past the M checkpoint, metaphase checkpoint, before they're entirely ready to do so, we could see the chromosomes going off in the wrong directions, right? The chromatids might go squirrely. I mean, if both chromatids go in the same direction, then we're going to end up with both chromatids in one daughter cell and no chromatids in the other daughter cell, which is going to be a huge problem, right? And so, you could say, well, if there's a problem here, that would lead to definitely the wrong chromosome number. Right? That would be one outcome. In other words, a form of mutation. Okay? If we're to allow the cell to begin mitosis before those chromosomes are really ready, right? In other words, if we were to start to initiate the thickening of the chromatin, that could also result in things that are going awry, potentially mutations. Right? And the thing about this is that mutations are actually one of the ways in which we can detect problems with the checkpoints, and we'll get into this future. But here's the thing. I mean, if you look at, not surprisingly, if we have cells that are undergoing continual cell divisions, that's going to give us faulty cell divisions and these mutations. And so one of the things that you can do to recognize one of the characteristics of a tumor cell, right? If you collect the genome out of a cancer cell, you look at the genome, you're not going to find the normal genome with just a single change. You're going to find dozens of mutational changes in the genome of the cancer cell. Okay? And that's completely understandable because we actually see mutations resulting as a consequence of bad cell behavior, right? And so the idea here is that badly controlled cell divisions can lead to mutations. And this is one reason why cancer cells are going to have mutations. That makes kind of sense to you, right? But the other thing is that one of the causes of aberrant behavior, of the kind of behavior that causes the normal cell to convert into a cancer cell, is a mutation itself. In other words, it could be a mutation in the control at that G1 checkpoint that leads to these excessive cell divisions in the first place, right? So in other words, mutation can actually cause a cancer. In fact, sometimes we require multiple mutations as a cause of the cancer. And we also see additional mutations cropping up as a result of the cancer's behavior itself, right? And I know we're kind of jumping ahead of ourselves. We haven't really talked about the genes in the level of detail that's required for us to have a really complete discussion of what mutations are. But at this point, you think of mutations in the most generic sense. It's some type of change. Right? We see genetic changes in cancer cells, and some of those changes might actually be the cause of the cancer's behavior. But once the cells start behaving incorrectly and having poor control over the cell divisions, that's actually going to lead to additional mutations, which might potentially make the cancer even worse, right? And so that's one thing to keep in mind. I guess the question is, well, why is it that cancers begin in the first place? It can be as simple as some type of problematic aspect in the control of the cell cycle. Okay. Also, pretty significantly, okay, sometimes cancers are caused by viruses. Right? Viruses do this because of what happens is that a virus takes up residence in one of your cells. Right? So this is your cell. Okay. And then inside of your nucleus, there might be a hitchhiking gene, which is the gene of the virus. That was not what I wanted to do. I wanted it to be in green, right? So yeah, you've got a whole bunch of your genes here in the nucleus, and these are your genes, blue ones. Okay, lots of chromosomes, but also there in your nucleus is the viral gene. Now, what's going to happen if your cell starts to behave incorrectly? In other words, if you start to have uncontrolled cell divisions. Okay. Your cell divides uncontrollably. As your cell divides, the viral gene is being replicated as well. So in other words, at the end of the process, you could have hundreds of virally infected genes. See that? Okay, now this is something that you want to, I bring this up because it's just too awesome to understand how this happens. Now, if you are a virus, right, you have to think like a virus. How could I make the most out of my viral infection, right? Most viruses might come in infect a host cell and then cause the host cell to make a whole bunch of new viruses, right? What this virus is doing is it's coming in, infecting host cell, and then causing the host cell to turn into hundreds, maybe thousands of viral infected cells. And then we could start producing the viruses, and we've just dramatically, enormously increased the total yield of viruses. The virus's replication, the virus's fitness, Darwinian fitness, has increased tremendously as a result of this behavior. So in other words, the viral gene, if it's causing a cancer behavior, is totally going to have an advantage relative to a viral gene that does not cause the host cells to increase in number, right? And if you think about that, the successful viruses are the ones that produce the most viruses. That's natural selection. And it kind of, it should not surprise you at all that it's not too uncommon of a thing for viruses to actually be the cause. The viruses can bring in to your host cells, to your cells, the genes that cause you to lose control over the cell cycle. All right. Now, since I've already let the cat out of the bag when it comes to genes, genes that normally make the G1 checkpoint behave well, but if we change those genes, G1 checkpoint doesn't do what it's supposed to do. The viruses can carry genes that make the cell behave badly. I want to give you like this concept of hope, right? It might seem pretty hopeless at this point, but we actually do have really sophisticated tumor suppressor mechanisms, right? And yes, they are under control of tumor suppressor genes. But the idea here is that we've got other safeguards that can detect bad behavior in the cells. And when they detect the bad behavior, they can stop it, right? So maybe you have a mutation that caused your cells to not behave properly. They don't follow the rules and they don't go into the G0 phase at the G1 checkpoint. Right? Your cells can tell, okay, well, this cell is behaving badly. I want that cell to either stop dividing, and you could just arrest the cell and say, okay, that's enough for you. You're not allowed to divide anymore. Right? Or we have a more spectacular form of control of these badly behaving cells. We've got like a cellular self-destruct button, right? Programmed cellular death. And it's got a name, it's called apoptosis, right? Apo meaning way, tosis, starting with a silent P, being the, I forget what that means, but you can look it up, right? And so, yeah, all hope is not lost. This happens, these kinds of things happen actually all the time. But because we've got mechanisms like the tumor suppression, we don't actually see tumors popping up all the frickin' time, right? Does that make you feel better? It should, until you realize these tumor suppressor genes might also mutate. And the mutation there would be really bad because, well, you know what? If you lose your ability to suppress the tumors, then you would be getting a whole bunch of cells that are converting into cancer cells without the opportunity to convert them back or to cause them to self-destruct. All right. Now, I know I'm kind of anticipating giving you a little bit of preview of discussions we'll be having later, but I did want to bring this in right now because otherwise it's kind of hard to understand why we really even care about the cell cycle and cell divisions, right? This is why.